Every movement, every memory, and every behavior is driven by a continuous flow of electrical and chemical signals within your central nervous system. While electrical impulses travel rapidly down the length of a single nerve cell, the actual communication between these neurons happens across a microscopic gap known as the synaptic cleft. To bridge this gap, your body naturally produces neurotransmitters. These are endogenous chemicals, substances manufactured internally to maintain your baseline physical and mental functions. When the production of these internal chemicals fails, the macroscopic effects are severe. If the specific neurons that produce dopamine degenerate, the lack of this chemical across the cleft causes the severe motor tremors characteristic of Parkinson's disease. Similarly, if acetylcholine-producing neurons degenerate, the resulting chemical deficit severs pathways required for memory retention, leading to the cognitive decline seen in Alzheimer's disease. Your ability to function as a healthy, autonomous human relies entirely on the precise microscopic balance of endogenous chemicals interacting at the synapse. We can intervene in this delicate environment by introducing external molecules into the body. These are exogenous chemicals, artificial substances ranging from engineered medicines to recreational drugs. This diagram shows a molecular comparison between natural and artificial chemicals. Though structurally simple, exogenous molecules dictate complex brain circuits by perfectly matching the binding sites of your endogenous systems. These synaptic hijackers fall into two primary categories: agonists and antagonists. An agonistic drug amplifies or mirrors the natural signal of a neurotransmitter. An antagonistic drug does the exact opposite. It blocks or completely silences the endogenous signal. We will analyze 4 specific exogenous chemicals to map exactly how their micro-level mechanisms dictate macro-level realities, causing effects ranging from temporary euphoria to total paralysis and profound analgesia. A drug's systemic impact is governed entirely by its targeted intervention at the synaptic cleft. This animated cross-section maps the mechanics of a healthy synapse. Due to a stimulus, the presynaptic terminal at the top releases dopamine into the cleft. These molecules temporarily bind to postsynaptic receptors at the bottom, creating a short-lived sensation of pleasure. This sensation ends when the dopamine molecules are quickly reabsorbed. They are recaptured into the presynaptic neuron through specific channel proteins, resetting the entire cycle. Amphetamine disrupts the cycle aggressively. Once introduced into the presynaptic terminal, it forces a massive, unnatural exocytosis, dumping an overwhelming wave of dopamine directly into the synaptic cleft. Because the cleft is completely flooded, the transmitters bind continuously to the receptors. The resulting sensation of pleasure is highly amplified and lasts for a significantly longer period of time. Cocaine also acts as a dopamine agonist to prolong pleasure, but it utilizes a completely different mechanistic strategy. Looking at the reset synaptic model, we see that cocaine does not increase the exocytosis of dopamine. Instead, it acts as a physical plug, firmly blocking the recapture channels on the presynaptic membrane. Dopamine is released in normal quantities, but it cannot be reabsorbed. Trapped in the cleft, the transmitters bounce off the blocked channels and repeatedly strike the postsynaptic receptors. Both amphetamine and cocaine act as dopamine agonists. One weaponizes the release mechanism, while the other weaponizes reuptake inhibition. We see similar hijackings outside the brain, specifically at the motor end plate, where neurons connect directly to skeletal muscle fibers. Under normal conditions, a neuron releases acetylcholine, an excitatory endogenous neurotransmitter. It binds to the muscle's receptors and triggers contraction. This visualization illustrates how curare exploits this pathway.
Curare possesses a spatial configuration highly similar to acetylcholine, allowing its molecules to occupy the specific binding sites on the postsynaptic membrane. Curare binds to the receptors, but it does not activate them. It merely occupies the space, preventing the actual acetylcholine from binding and passing its signal through. Because the natural excitatory signal is physically blocked from reaching the muscle fiber, the muscle itself cannot contract. The fiber goes completely slack. By perfectly mimicking a neurotransmitter's shape without adopting its function, curare operates as a pure antagonist, turning a microscopic receptor block into total paralysis. This diagram maps a nociceptive or pain circuit. Processing pain requires a more complex synaptic architecture, involving a presynaptic terminal, a postsynaptic terminal, and a third modulatory neuron attached to the side. When you experience a painful stimulus, the presynaptic neuron releases P substance. This analgesic neurotransmitter crosses the cleft, binds to the postsynaptic neuron, and induces the sensation of pain. To regulate this, the third modulatory neuron releases its own endogenous chemical, a natural analgesic called enkephalin. Enkephalin binds back onto the presynaptic neuron. This partially inhibits the exocytosis of P substance, temporarily dulling the pain signal. This natural relief is inherently brief. Specific bodily enzymes quickly digest the enkephalin, removing the inhibition and allowing the pain signal to resume. Morphine hijacks this specific control mechanism. Because its spatial configuration is highly similar to enkephalin, morphine acts as an agonist to this modulatory system. Once injected, the exogenous morphine navigates to the presynaptic neuron and binds to the exact same receptors used by enkephalin. Here, the body encounters a biological blind spot. There are no specific enzymes capable of digesting the exogenous morphine molecules. Because it cannot be broken down, the morphine completely and permanently blocks the exocytosis of P substance. Morphine achieves profound, long-lasting analgesia by acting as an unstoppable agonist to the brain's natural, endogenous inhibitory system. This split screen reviews the mechanistic toolkit of the hijacked synapse. Exogenous chemicals overpower the system by amplifying exocytosis, blocking reuptake, utilizing competitive binding, or resisting enzymatic degradation. This leads to a clinical question. What happens to the human organism when these high-magnitude chemical perturbations become chronic? The central nervous system is highly adaptive. When faced with continuous, massive flooding from exogenous drugs, it interprets the event as a toxic chemical imbalance and attempts to correct it. This diagram illustrates tolerance at the biological level. As the cleft is repeatedly flooded, the organism physically adapts by downregulating its own hardware. The postsynaptic membrane deliberately absorbs and removes its natural receptor sites to protect against overstimulation. Because there are now fewer operational receptors, the exact same amount of the drug yields a significantly reduced effect. This structural adaptation forces the user to continually increase their dosage just to cross the new baseline and achieve the original chemical effect. If consumption is suddenly interrupted, the system faces physical dependency, a catastrophic biological failure. With the exogenous drug abruptly gone, the downregulated system is exposed. There are barely any natural receptors left, and insufficient natural neurotransmitters to bridge the gap. The required neural signals flatline. This sudden chemical vacuum triggers severe physical pain, psychic distress, and systemic physiological breakdown across the organism. Driven by the psychic memory of chemical euphoria, the individual must navigate the agonizing physical reality of a starving, structurally altered neural network. Clinical addiction is a measurable state of physical dependence, forged directly in the microscopic machinery of the synapse.